Okay. So, so now we have Benjamin Siranosian from Harvard University.
Hi, everyone. My name's Ben. I'm currently a PhD student at Stanford, but today I'm talking about some work that I did last year when I was at the Broad Institute, affiliated with Harvard, definitely. First off, I wanted to thank the people that actually helped me do this work. First, Yuri Ben David is a postdoc at the Broad, advised by Todd Golub and Ramin Brukim. He really took on this entire project and coordinated the effort of many different labs on the wet lab and the computational side. So he deserves a huge acknowledgement for this. And as you can see, this was a major collaboration between many different groups. I received funding from the ISMB to travel to Chicago to present this work, and also from Stanford, and the actual research was funded by the Broad Institute. So today I'm going to be talking about genetic and transcriptomic instability in cancer cell lines and how that influences things like drug response. Cancer cell lines are a major keystone of biomedical research today. However, many of the findings from large-scale drug screens or compound identification can't be reproduced. And a recent publication found actually about 50% of preclinical research is not reproducible, part of that due to instability or differences in cell lines that they've used. And so today I'm going to talk in 3 different sections. First, how variable are cancer cell lines or cell lines obtained from different sources that we think are the same? Does this variation actually matter? So is it just a curiosity, something we should study, or is it actually an implication for cancer research? And is it all bad news? Is there anything that we can do about this moving forward? So kind of get the conclusions out there at first. Cancer cell lines obtained from different sources, different labs, and from different years are very variable. And this is maybe not a surprising finding, given that cancers are known to evolve very rapidly and their genomes are already very messed up. This variation does matter. We show it matters for the response to a number of different drugs. And we don't think it's all bad news. You can actually use a cell line panel like we describe as a system of isogenic cell lines. And we think that there are a few different ways that you can cheaply and quickly characterize the cell lines that you're working with in your lab to kind of prevent the findings that I'm talking about. So I'll just kind of go quickly through the data because I don't have too much time, but this is the wide-scale genetic and transcriptomic variation that we found. We started with 27 isolates of the cancer cell line MCF7, which is known to be a breast cancer estrogen receptor positive cell line. These came from many different sources over, I think, a span of 11 different years. We actually had one really interesting collection where the same lab provided us cell lines from 10 years that they took from their freezer. These were subject to freeze-thaw cycles, but not serial passaging. So this is actually the most cohesive set of cell lines that we obtained. We also had a few cell lines that had supposedly neutral genetic manipulations, such as introduction of GFP or Cas9 for CRISPR testing. So first off, we saw very wide copy number variation via low-pass whole genome sequencing. Not unsurprisingly, around 25% of the genome was altered in these cell lines. However, there was a really heterogeneous set of copy number gains and losses across the entire population. So we had 20% of the alterations which were common to all strains, 10% which were private to an individual strain, and the rest were really variable. And as you can see on this heat map on the right, where red indicates gains and blue indicates losses, it's pretty heterogeneous. We also saw an interesting clustering where the cell lines that we know to come from the same source over many different years, that lab that I talked about, they all cluster together. That's pretty much what we expect. If we look at the mutational landscape, we see something similar, where some mutations are shared by all lines, some are private, but the majority are pretty heterogeneous. And we see a very similar clustering, where the cell lines obtained from the same source appear to be very consistent. We also measured transcript levels directly of 1,000 genes with the L1000 assay. We then inferred the level of the rest of the transcriptome via an algorithm published by a group at the Broad. And what we saw was that although these MCF7 strains appear to be very different, They all cluster closest to MCF7 and other breast cancer cell lines that we've profiled in the past. So we don't think things are too far away from what you would expect. However, we did see a high number of differentially expressed genes when we compared pairs of these strains. So by now, I hope I've convinced you that we've seen wide-scale genetic and transcriptomic variation in these 27 isolates of MCF7, but does it actually matter? As an example, MCF7 is known to be estrogen receptor positive, and it's used as a model for that type of cancer research. If we look at just the copy number of ESR1, which is the estrogen receptor gene across our panel, we see pretty wide variation. And we also see that the copy number is correlated with the expression of this gene. So if you're using MCF7 as a model for ER-positive breast cancer, you expect it to have the gene and express it. And we've shown that's really not the case. So you need to kind of confirm this if you're using this as a model. We also looked at the impacts this genetic variation may have on drug response. So we took over 300 compounds and we treated these 27 cell lines at a single dose and measured the viability change of these lines. We saw 55 compounds were active in at least one strain, and that's the heat map I'm showing here on the right, where dark blue indicates a significant growth inhibition. However, of these 55 compounds, 48 had at least one strain that was entirely resistant, meaning that it received less than 20% inhibition of growth. So you can understand why this would be important if you're screening drugs in different cell lines to try to identify something that is maybe a targeted therapy for cancer. To validate this further, we subjected a few drugs to 8-point dose response testing across the 27 different strains. Here I'm just showing one drug, bortezomib, which is a proteasome inhibitor. And we had several lines that were resistant and sensitive to this strain, as you can see by the graph showing viability versus concentration. We also saw several interesting cases that I can talk about further later, where drug response was correlated to expression of the genes in a particular pathway that drug is known to inhibit. And same thing with mutations. So to kind of wrap things up, we wanted to know if this was a fluke in MCF7, the cell line that we use, or is this a feature of cancer cell lines more generally? We repeated the same experiment in A549, which is a lung cancer cell line, and we saw similar levels of genetic and transcriptomic variation within a number of different clones of A549. We also took it a step further thanks to a reviewer and did simple sequencing of 11 different cell lines, multiple strains, and we saw that the levels we observed for MCF7 and A549 are pretty similar to what we see from other cancer cell lines and also human non-cancer cell lines. And really, what drives the genetic variation that we saw? And the answer to that is serial passaging of cancer cell lines, right? As you passage these lines and they divide multiple times, they're becoming more and more distant from each other. We actually saw that freeze-thaw cycles, the cell lines that we got from that group that I talked about earlier, don't really affect genetic variation too much. However, introduction of GFP or other neutral genetic manipulations are pretty bad for cancer cell line instability. And is it all bad news? I like to say no, because we actually found that a strain panel like this for MCF7 is basically an isogenic panel of cell lines. And it's actually better at identifying the targets of known drugs, the mechanisms for known drugs, than a panel of cell lines itself, because they're so close genetically. They're only altered in a few spots. So moving forward, we kind of have three main conclusions from this work. The first is that there are obviously inconsistencies in genetic and drug response for different isolates of the same cancer cell line. We think that researchers using cancer cell lines really need to confirm the identity of what they receive, whether it's from a commercial vendor or from a neighboring lab. And I think you need to recharacterize the genomic features of a cell line you're working with after several passages or after introduction of some GFP or something like that. Fortunately, we found that low-pass whole genome sequencing, a pretty cheap and efficient method to characterize cell lines, is well correlated with all the other metrics of distance that we had. So that's an easy way to repeatedly check what you have going on. And we published a tool to use low-pass whole genome sequencing to measure distance between your data and other data. And with that, I'd like to say thanks for listening, and thanks again to the people who helped me out with this project.
Okay, so we have time for one guest question. Yes.
Very nice talk. Did you check the levels of methylation in the expression of your genes?
No, we did no methylation.
Because methylation could affect the expression of your genes.
Absolutely, yes.
So thank you again.